This is Blood and Cancer, the official podcast of MDedge Hematology/Oncology, where we discuss the latest research, treatment considerations, and other issues related to hematology and oncology. Welcome to this podcast. I'm your host, Dr. David Henry. And of course, we're recording this in January 2021, and the major issue of the day a year later continues to be COVID and how it might affect oncology and then HIV, hematology. And I'm delighted today to be speaking with Dr. Drew Weissman, who is physician, professor of infectious disease at Penn's Perelman School of Medicine, and has quite an interesting history with regard to what we're now using in messenger RNA vaccines. So Drew, welcome to this podcast. Thanks for taking the time to speak with me today.
Oh, of course.
So I thought we'd begin, you're so modest, see if I can pull out of you, because you know I've heard you do this before, it's just wonderful. The brief history of this type of vaccine, which we're now using in the Pfizer and the Moderna, of messenger RNA and your role in helping to develop that. So how did that play out?
So I think what people have to understand is that the first time mRNA was used with an idea of using it as a therapeutic was in 1990. At that point in time, it was used for vaccines, but nothing else. And the reason for that was that the RNA was highly inflammatory. So when you injected it into an animal, the animal got sick. That's where Katalin Karikó and I started studying around 1998. And what we wanted to understand is what made the RNA, it's a natural molecule, what made it so inflammatory to the host. So we studied that for a number of years. We identified a variety of receptors that recognized RNA. But the big turning point came when we figured out how to make the RNA non-inflammatory. That was in 2005. And that's where we put what are called modified nucleosides. So our cells and everything from bacteria through us modifies its nucleosides. There's over 100 different types of modification. They're naturally occurring. So what we did is we put some of these modified nucleosides into the mRNA. And when we did that, we found some that got rid of the inflammation and, more surprisingly, made more protein from that RNA. And the increases ranged up to about 1,000-fold in mice. So what we were able to create was an RNA that was well tolerated, that didn't induce inflammation, and made a lot of protein. And that's the basis for Moderna and for this vaccine.
So I understand, to say it again for my clinical mind, we have cells, factories all through our body, all as humans, and we're being told by the DNA to RNA from our nuclei what to do. So you found a way to inject it. It gets to those cells, doesn't need the nucleus, just goes to the cytoplasm, talks to the ribosomes, if I remember my biochemistry, and they just start cranking out this protein of choice. In this case, I think it's the COVID spike protein, yes?
Yes, exactly.
Okay. So I think I've heard you say that, you know, that you get the flu shot, it lasts for, in terms of antigenicity in your arm, for maybe hours to a day, and then it turns on immune response real quick and goes away. Here with these mRNA-based vaccines, you're making tons of this spike protein for weeks after a single shot.
Yeah, so that's really critical in immunology. And what we understand is that to make a good immune response, you want to have antigen present for a long time. And we don't know how long is too much, but we designed the RNA LNPs that will make protein for 10 to 14 days. And we found that that results in really a great immune response. There's a second component that people don't understand, and we still don't know the mechanism, but most vaccines have an adjuvant. That's something that stimulates the immune response. Most adjuvants induce, they're called Th1 or Th2 immune responses. The LNP that's used in this vaccine is an adjuvant, but it makes a specialized CD4 helper. And the only thing this CD4 helper does is it drives antibody production. It increases antibody affinity, it matures the antibodies, it makes long-lived plasma cells, so that gives you long-lived antibody responses. This is the only adjuvant that we or anybody knows of that specifically induces these particular helper T cells.
So if we're producing all this antigen spike protein for weeks after an injection, people think about their reaction to the shots, Pfizer, Moderna, 24, 48 hours. Couldn't we have reactions as long as 10 days to 14 days out?
Yeah, so actually the reactions are the lipid nanoparticle. And what's
Oh,
happening is that the adjuvant effect of the lipid nanoparticle is what people feel. That's what gives you the arm pain, the flu-like syndromes, the fevers. Once that's gone, which is usually by 24 hours, that the symptoms are gone. The
ah,
spike protein itself, when we say produce for 14 days, the amount produced drops rapidly. So you don't get a huge amount of protein over 14 days. You get a lot of protein in the first day, and it slowly decreases over 14 days.
okay. All right. So a couple practical questions there before going on to HIV and oncology. We hear don't take an Nsaid. Maybe you could take Tylenol, Benadryl. So to prevent that LNP reaction and not impair immunity response, do you agree with that? No NSAIDs.
So the answer is we don't know. We
Okay.
just haven't tested it yet. We do know that with flu vaccines, taking an NSAID with a flu vaccine reduces your response. So our assumption is that similar will happen with this vaccine, but it hasn't been tested. We just don't know.
Okay. So as you record this again in late January 2021, we're hearing about variants, which to me makes perfect sense. A virus which proliferates in nature around the world is going to, by random chance, mutate. So just to name them, we're hearing Brazil, South Africa, California, UK. What can you say about what is known about those and whether or not the vaccines will address those?
So I think what people have to realize is that a year ago, probably November, this was a bat coronavirus. And it was only in bats. It had been in bats for probably thousands of years. And it suddenly made the jump to people. So as far as the virus is concerned, it wants to figure out how to better infect people. So I don't really call these mutations. I think they're adaptations. These are the virus figuring out how to infect human cells. And it's completely expected, and it's going to continue to happen. There's a number of variants that have appeared. They've been appearing since really March was the first time we saw the first variant. The big issue is there's really two kinds of variation. There's variation that happens when a virus tries to figure out how to better infect somebody. And then there's variations where the virus tries to avoid immune responses. So influenza mutates every year, and those adaptations are to avoid the prior year's immune responses. With COVID-19, most of the variants that have been seen are equally addressed by the vaccines. The South Africa one is the one that we know has reduced. I think for the Moderna, it's about 1/6 of the response. We don't know if that's because the virus wants to avoid an immune response or this adaptation made it grow better. The issue is what do you do with these? Are we going to be making coronavirus vaccines every year for the variants that appear? Coronavirus mutates very slowly. Influenza, HIV, HCV mutate very rapidly.
Mhm.
So we don't know the answer to that yet. It's easy with the RNA vaccine to plug in the mutant or any mutant to make a booster or to make an improved vaccine. Moderna is working on that now. BioNTech, Pfizer is doing that. We're doing that with our Thai vaccines. Um, so it's going to be catch up. To me, the big issue is how long will it take to vaccinate the entire world? because until we do that, more variants are going to appear.
Yeah, just a natural biology evolution of the
Yep.
virus. Okay. All right. Well, let's turn to some of our focus in oncology and HIV. So, the oncology patient is getting chemotherapy. That's typically a myelosuppression, neutrophils, not so much other cells, and or our burgeoning field of checkpoint inhibitors, the PD-L1 ligand or PD-L1 receptor inhibitors. Anything you can say about what we know and whether the vaccine will respond equally well or not in patients, cancer patients getting those treatments?
Yeah, so the issue is we don't know. There haven't been any clinical studies. There really haven't been any animal studies on animals receiving chemotherapy. Some things that we take from other vaccinations. So we know that when we knock down the immune system with chemotherapy, it doesn't respond as well. Vaccines don't work as well. I would expect the similar with the COVID vaccines. The issue is, when do you want to give the vaccine during a chemotherapy cycle? And that's another unknown. Typically chemotherapy knocks down myeloid cells 7 or so days after the dose. They hopefully come back up by day 28 when they get the next cycle, or whatever the timing is with the cycle. If I were to suggest when to vaccinate, I might wait until day 14, because the immune system, the germinal centers where the B cells are produced, forms about 2 to 7 days after the vaccine. So there, your cells are coming back. And what I learned in my oncology days, which were a long time ago, is that cells coming up are always better than cells going down for their immune responsiveness.
Quite true, quite true. And same answer for the checkpoint inhibitors, because I suspect we don't have data, but what would you theorize?
So that they're different. So what a checkpoint inhibitor does is it boosts the immune response by altering your cells that are overstimulated and are non-responsive. So what a checkpoint inhibitor does is it boosts the immune response. So that, the worry is, could you make too good an immune response against the COVID vaccine? We don't know the answer to that. But my fear wouldn't be that they'd get a worse immune response. It would probably be the same or potentially boosted. With influenza, I heard that PD-L1 usually gives you a similar immune response. About a quarter to a third of people have a somewhat enhanced response.
Okay. Well, let's just for fun add one more variable to what we do and don't know, and that's the HIV patient. As you know, we've come a long way since this epidemic started, and most patients now who take their medication, usually integrase inhibitor based, have robust CD4s and undetectable viral loads. They're never quite immunocompetent like they were before, but close. So would you expect them, based on that immunology, to do same, better, worse with vaccine?
So if you've got a patient who has no viral load and CD4 counts above 200, they should respond well. Not as well as control patients, but they should respond well. And the vaccine is powerful enough that I think their immune response will be protective. For patients with high viral loads who don't take their antiretrovirals and very low CD4 counts, that's more of an unknown. And certainly the vaccine is not going to work as well. How poorly it'll work isn't really known.
Okay. Now, there's been some talk, of course, meetings are continuing. We're Zooming all the time. Our national hematology meetings were in San Diego, but they were Zooming. And word came out about our CD20 antibodies, of which there are many, rituximab being one of the first, and there are others in B cell malignancies, for example. And they might blunt the B cell response of vaccination. What can you say about that?
So what I know of rituximab is that it depletes all of the B cells in the circulation. You will still find some B cells in tissues and lymphoid organs, but not a lot. They typically will deplete for 7, 9, or so months. In the absence of B cells, you're not going to make a good antibody response. What we don't know is that these vaccines also make T cell responses, and rituximab has a minimal effect on those T cell responses. We believe that they're going to be protective. They're probably not going to prevent infection. They're probably going to modulate the infection, but we don't know how much. We haven't tested them on mice that don't have B cells. So we don't know how good that T cell response will be in protecting against serious disease.
Okay, I think a guideline for our listeners that we're getting from our heme and onc mother organizations is if you have to have your CD20 antibodies, so be it. If you can do without it for a while, maybe better, given what we do and don't know. Well, I have a little list of a couple of practical things that come up, and I'll just name them. Vaccine and immunity, if you get one instead of 2 shots for whatever practical reason. If you've recently had COVID, should you still get your shot? You want to take those 2 scenarios.
Yeah, so again, we just don't have a lot of data. We should soon. I think over 20 million people have received the mRNA vaccine. And strikingly, it's been incredibly safe in those 20 million people. The worst side effect we see is an anaphylaxis, anaphylactoid reaction in less than 1 in 100,000 people. But the issues with severe immunosuppression in taking this vaccine are unknown. Maybe you can give me each question individually and I'll address it.
Yeah, so we're seeing this now, patients recently had COVID infection. They were positive with or without symptoms, and now they were planned to get their shot. Should they still get their shot, say 2, 3, 4 weeks after knowing COVID positive?
So what clinicians who do a lot of vaccinations are saying is they're waiting upwards of 90 days. And I think they're doing this for 2 reasons. The first is that COVID infection in 95+% of people gives you protective antibodies that will last at least 3 months. The second is that we've seen that people who have recently had COVID infection have more severe adverse reactions, more arm pain, swelling, fever, flu-like symptoms. So it's basically, it's safe to wait and patients will tolerate it better. I'm sure the vaccines would work well 2 weeks after somebody has had COVID. It might work really well because it'll boost an ongoing immune response.
Makes sense. Now, thinking down the road, here we are early 2021, hopefully a large percentage of the world will be vaccinated as we go through this year. And people may say, well, how good's my immunity? As you know from the clinical trials, the placebo controls, it was 95% didn't get severe infection, but how immune were they? I suspect those data will start to come out. Do you think we will be checking antibody status? Any role for that you can envision down the road?
So what the phase 3 trial showed, and this is both Pfizer and Moderna, is that the titers, the level of neutralizing antibodies, were higher than patients who had recently been infected. So the vaccine gives a better immune response than infection. The issue that we don't know, and this is being studied in both phase 3 trials and in other trials, is what's the durability of the response? And that's really the critical thing. So with tetanus lasts about 10 years unless you've stepped on a nail, where you say 5 years. Smallpox lasts your entire lifetime. So vaccines have different durabilities of protection. For mRNA LNPs, we just don't know what that is in humans. We haven't followed them for long enough. So both of those studies are doing quantitative antibody neutralization assays over time to see how long the response lasts and when we need to boost people. The problem for clinicians in general is that The antibody assays available, many of them are directed at the N protein, not the spike protein. And the vaccine only has spike, and they're not quantitative. So they'll tell you, yeah, you've got an antibody, but they won't tell you how high it is and whether it's protective.
Okay. Now, the mRNA vaccines, they're on the market, at least in the US, they're being given. We hear the soon to be a J&J, Johnson & Johnson vaccine. Can you comment what's their mechanism of action? What might we expect differently? And I think it's one shot.
Yeah, so the J&J is an adenoviral virus. Adenovirus is a common cold virus. And what they've done is they've inserted the spike protein into the genome of the adenovirus. What we're waiting for, and according to Tony Fauci, J&J will submit to the FDA in the next few weeks what the efficacy is. So we've seen a few different efficacies. Both RNA vaccines were about 95%. One of the Chinese inactivated vaccines was 50%. The Russian vaccine was 93%. The issue is they published that before they did the phase 3 trial. So people don't really trust that number. And the AstraZeneca vaccine, where they screwed up the clinical trial, they came away with an efficacy of 70%. But we have to take that in mind that they did two different vaccines. One was a full dose followed by full dose. The second was a half dose followed by a full dose. The full dose vaccine had about 62% efficacy. The half dose, they claimed had 90% efficacy, but the numbers were so small that the FDA wouldn't accept that. So what's being given is full dose, and a reported 70% efficacy. We have to see what Johnson & Johnson's efficacy is. If it's 90, 95%, then the whole world is going to use it. If it's 60%, people are going to think.
Okay. Well, I think I'll bring our wonderful discussion to a close and just summarize for our oncology, hematology, HIV patients. We're hearing when you're getting chemotherapy, maybe wait for those cells coming back. I think I learned the same thing in oncology as you did. They're a little better, healthier when they come back as when they're going down. So wait till your nadirs coming out of it. The checkpoint inhibitors might, for interesting reasons, even work the same, if not better. And then the HIV patient is well controlled. You would expect nearly the same result, but be careful with that HIV patient who's not taking medicine, viral load high, might not respond as well. And I think that'll do it. I want to thank you, Drew, for taking the time to speak with us today and congratulate you for your instrumental role in bringing this particular technology of messenger RNA along to where it's helping to save the world. So can't thank you enough. We've been speaking with Dr. Drew Weissman at the University of Pennsylvania Perelman Center, where he is a clinician scientist and professor of infectious disease. Drew, thanks so much for speaking with us today.
Thank you very much.
Blood and Cancer is produced by MDedge. Our editor is Jen Smith. All MDedge podcasts are produced by executive editor Kathy Scarbeck, and I'm your host, Dr. David Henry.